Hello everyone, my name is Leah Eversole and welcome to my mini lecture on neural transmission. First we will start with the parts of the neuron. There are other types of cells in the nervous system, but the most important cells are the neurons. These neurons have characteristics, three of which are excitability, the response to stimulus, conductivity, ability to conduct electrical signals, and secretion, the release of neurotransmitter in response to conductivity activity. The parts of the neuron are the dendrites, which receive signals from other cells, the cell body or soma, which organizes and keeps the cell functioning, and this houses the organelle, the nucleus, which controls all the functions of the neuron. The cell membrane protects the cell, and the action potential, which we will learn about later, of the neuron is initiated in the axon hillock. Then we have this long process called the axon, which transfers the action potential signal to other cells and organs. This is covered by a myelin sheath, which increases the speed of this action potential signal. And in between these Schwann cells that produce the myelin sheath, we have the nodes of Ranvier, which allow for the diffusion of the ions in this process of the neurons. And this is saltatory conduction when the action potential jumps from one node of Ranvier to the next. And at the end of the axon, there is the axon terminal, which forms the junctions with other cells to communicate to these cells. This is a multipolar neuron, this just meaning that there is one long axon and many dendrites on the cell body. Next we have the electrical portion of the action potential. This is a resting membrane of a neuron. It is negatively charged on the inside at about negative 70 millivolts. And on the outside it is positively charged. As you see, there are cations, meaning positive ions, such as sodium and potassium. And here is how the electrical conductivity works. We have the resting membrane potential at negative 70 millivolts. Then, when it reaches threshold at about negative 55 millivolts, it depolarizes, and this occurs when threshold is reached. And voltage-gated sodium channels open, and sodium enters cell rapidly, reversing the polarity from negative to positive. Repolarization occurs due to the closure of the voltage-gated sodium channels and opening of the voltage-gated potassium channels. Potassium moves out of the cell and polarity is reversed from positive to negative. Hyperpolarization. This occurs when the voltage-gated potassium channels stay open longer than the time that is really needed, and this makes the cell even more negative than the resting membrane potential at about negative 80 millivolts. Next we have the chemical portion of the action potential. Here we have a presynaptic cell and a postsynaptic cell, and in between is the space called the synapse. On the postsynaptic cell there are receptors, and here we have little packages called vesicles that carry neurotransmitter across the synapse to these receptors on the postsynaptic cell. And neurotransmitter is broken down within the synapse, or a process called reuptake on the presynaptic cell occurs to terminate the use of this neurotransmitter. And as you can see here again, in the transmission segment of the release of a neurotransmitter, it goes from the presynaptic knob over to the receptors. This could be on a muscle or on another neuron or gland. This slide shows that there are many synapses on the dendrites of the postsynaptic neuron, and these are constantly firing signals, some of them inhibitory and some excitatory. As you can see, all of the red are inhibitory and the green are excitatory. And all of this equals summation, which occurs in the axon hillock, and when that sum is equal to negative 55 millivolts, approximately the threshold, then an action potential is conducted down the axon of this postsynaptic neuron, and the communication between the cell carries on from there. Thank you for watching, and have a great day. Goodbye for now.